Good morning. Today we are going to discuss about types of bacterial culturing. First of all, we can look what is bacterial culturing. It is a method of multiplying microbial organisms by letting them reproduce in predetermined culture medium under controlled laboratory conditions. These microbial cultures are foundational and basic diagnostic methods used as a research tool in molecular biology. These bacterial culturings are of four types: pour plate method, spread plate method, and streak plate method. The streak plate method is again divided into four: continuous streaking, radiant streaking, T streaking, and quadrant streaking. First of all, we can look what is pour plate method. It is a method of preparing bacterial culture for enumeration and isolation purposes. Enumeration means for counting the exact number of bacterial cells and to isolate the bacteria. The medium is maintained in a molten state of 45 degrees Celsius and 1 ml of diluted microbial sample is forced into a sterile Petri plate and 10 to 20 ml of molten agar is added. And these all the contents we added above are mixed well by both in clockwise and in also counterclockwise rotation. This media is then allowed to solidify and incubate in an inverted position. So we are not doing it in a normal position. We just reverse the position of the plate. The advantages of this method is it will allow the microbial cell to evenly spread out through the entire medium. Next is spread plate method. It is a technique used to treat bacteria in suspensions. In this method, approximately 0.1 ml of sample containing microbes is added directly to the center of solidifying agar plate and evenly spread on the surface of the plate. Again, the plate is incubated in an inverted position for the required period, as we saw in case of pour plate method. Third one is streak plate method. It is a rapid qualitative isolation method where inoculum is streaked over the surface of other plate. Inoculum is the point of infection of a pathogen. Okay. So often another plate in such a way so it thins out the bacteria. So in this method it is not necessary that we should have a serial dilution. Okay. So serial dilution to perform as a proper streaking is not required in this method. We can directly isolate the bacterial colonies. And one of the most preferred methods for isolation of bacteria is streak plate method. The streak plate method is again classified into four. That is, as we have mentioned earlier, it is classified into four. That is continuous streaking, radiant streaking, T streaking, and quadrant streaking. So first we are going to discuss about continuous streaking. In continuous streaking, a loopful of inoculum, that means a loopful of infectious pathogens, are continuously dragged across the surface of the medium in a zigzag fashion. So this is continuous streaking. And in radiant streaking, a loopful of organisms is introduced on a small area near and it's just a plate. 7 to 8 radial streaks are then drawn out with this re-sterilized loop. So first of all, the loopful of organism is just introduced into an area, and then similar like that 7 to 8 radial streaks of them are again drawn out with the re-sterilized loop. So first we sterilize a loop. First we took a sample, and when we use it with the sterilization loop, after that for next sample we have to re-sterilize it. Next kind of quadrant streaking is T streaking, and this kind of streaking has three phase pattern. It divides the bacteria plate into three different sections, and this is in the form of a T. Thus the name came from this structure, as we can see in the diagram that it is in the shape of a T. and this plate is turned 90 degree and a re-sterilized loop is dragged through second section starting from previously streaked the portion continuing zigzagging pattern. So it is just a continuous way. So first of all, if we do a zigzag pattern from one position, continuation as a continuation to that, through the second section we started from the previous streak portion, and this procedure is repeated once more, being cautious. So there are, as we said, that there is three phase pattern, and after this second section we again do this once more, and this step is done very cautiously. In this method, each time the loop gathers fewer and fewer bacteria until individual cells are separated finally in the last streaking section. So we carefully isolate all these bacterial cells up to the final streaking section. The last kind of streaking is quadrant streaking. As the name suggests that it's a quadrant, so there would be four equally sized sections, and these four equally sized sections are straight down another plate. Inoculum is added to the plate with a charged loop which re-sterilize after each striking. So after so there were four strikings, right? So after each striking, this loop is again re-sterilized. All four streaks are streaked in the same pattern by spreading the microbes. That's all. Thank you.